Everyone, and thank you for joining us today as our group's project explores the effects of lead pollution on Arabidopsis thaliana, a model plant widely used in biological research. My name is Daisy Blasi, and I am presenting today alongside Infinity Woodruff, Nathaniel Goff, and Harrison Stuckey. Our goal with this study was to better understand how lead, a common environmental pollutant, affects both the visible traits and the gene activity of this specific plant species. As we move forward, here's a quick overview of what we'll be covering today. I will go ahead and start with the background information outlining why this research is important and presenting our research question. From there, Infinity will take over to discuss the methodology, detailing our approach to data collection and analysis. Next, Harrison will go ahead and discuss the expected outcomes, results, as well as his take on data collection and analysis using Python. Finally, we'll go ahead and wrap up with the significance of our research, followed by our conclusion and references with Nathaniel. In today's presentation, we'll be discussing how pollution from industrial, urban, and agricultural sources release harmful substances into the environment, including heavy metals like lead. These pollutants can significantly impact plant biology, affecting both the physical traits like root structure and pigment levels, as well as internal genetic processes like gene expression and mutation rates. Arabidopsis thaliana, a small flowering plant, is directly exposed to airborne and soil pollutants in its terrestrial environment. While some research exists, there are still major gaps in understanding how pollution affects plant systems, especially in terms of long-term resilience and how invasive species may respond. Our research aims to address this by asking how has lead pollution affected Arabidopsis thaliana. Now jumping into some background information. Pollution is a powerful environmental stressor. Industrial emissions, urbanization, and agriculture all release harmful compounds like heavy metals, phosphates, and volatile organic compounds into the ecosystems. Terrestrial plants like the Arabidopsis thaliana are exposed to these pollutants through both the air and the soil. It is this kind of exposure that can cause major changes in plant biology. Physically, it can alter things like stomatal density, root structure, and pigment composition. And on a deeper level, it can also trigger genetic responses such as shifts in gene expression or even accumulated mutations. We chose to look at this plant specifically due to the fact that many scientists consider the Arabidopsis thaliana a model plant species. This is due to the fact that it has simple genetics and a small genome, which when being fully sequenced makes it ideal for genetic studies. The plant also has a short lifestyle of about 6 weeks, allowing for rapid generation and results. It is easy to cultivate and can produce a large number of seeds, facilitating extensive experimentation. And lastly, its genetic simplicity and desirable traits enhance its value for genomic analysis and molecular genetic research. However, despite these growing concerns, there's still limited research on how pollution affects plants across different ecosystems, especially in terms of genetic changes, species resilience, and invasive species. With all of that information, this brings us to our central question. How has lead pollution affected the Arabidopsis thaliana? This study looks at both the physical traits, or the phenotypes, and the gene activity, or the genotypes, of Arabidopsis in response to lead exposure. By focusing on this model plant, we're aiming to uncover how environmental toxins impact biological function, and in doing so, provide insights that can inform conservation strategies, help predict how plant communities might respond to future environmental challenges, and allow us to understand the resilience of potential invasive species. Next, Infinity will go ahead and start explaining our methodologies.
Hi, my name is Infinity, and I will begin discussing the methodology for how we determine the effects of lead on Arabidopsis thaliana. So the first step of our NGS analysis methodology is to do alignment. So what is alignment? It is the aligning of short DNA or RNA sequences to a reference or a known genome. So before the actual alignment can be done, indexing needs to be completed. Indexing is when unique sequences are added to each DNA or RNA fragment. That way they can easily be identified from each other. So this indexing is done to ensure that efficient mapping to the genome is completed, and it was done within R using the build index function in R subread. So following the completion of indexing, we're able to actually do alignment. RNA-seq data was gathered in FASTQ format from NCBI. We needed this FASTQ format because it contains raw sequence data and quality scores. So these files were then used to align to the index reference genome that we just previously obtained. And this was using the align function in R subread. Our output were BAM files that contained mapped reads. Following alignment, we needed to do some type of quantification of our data. So what does this mean? We basically needed our NGS data to be measured in numerical terms. So our first step to get this data was to do some annotation. An annotation file in GTF format was acquired from NCBI. This GTF file, which also stands for a genome annotation file, features things like genes, transcripts, exons, and coding sequences. And the reason for this was because it defines genomic features using quantification. So following the gathering of this annotation file, we're able to actually count our reads. This was done using the feature counts function in R subread. So this was able to count the number of reads mapped to the annotated genome. And then our output from this was a counts table and the summary statistics. And more detail on what these reads actually are. We could also define them as the number of reads sequenced by a DNA or RNA fragment. So now that we had completed our alignment and our quantification, we needed to do some more complex analysis, which would be our differential gene expression analysis. So the first step of this was to simulate our replicates. The data that we had acquired from NCBI did not contain any replicates, which would be essential for a successful differential gene expression analysis workflow. So in order to make up for this lack of data, we decided to simulate them. So what are replicates in the first place? They are parallel measurements of biologically distinct samples that capture random biological variation. So in order to simulate these replicates, we decided to use the set.seed and rnbinom function, which would generate random count data following a negative binomial distribution that was suitable for RNA-seq data with some common parameters consisting of mean distribution and dispersion that are observed in real datasets. Following this simulation of replicates, we're able to create our metadata. Metadata is basically data that contains things like variables, titles, how samples were collected, number of samples. There's lots of different things that could be contained in metadata. So to create the metadata, a data frame with conditions and row names was created to correspond to the count matrix. So now that we had simulated our replicates and we created our metadata, we could finally create our DSeq2 dataset. So this dataset was created using the DSeq dataset from matrix function, which is pretty straightforward, by combining the count matrix and metadata with the design parameter as condition for the variable of interest. So now that we had completed our alignment, our quantification, and complex analyses like differential gene expression analysis, we could finally put together a methodology for our visualizations. So our first visualization would be the volcano plot. The data was prepared for this using the DSeq2 results table, which is then loaded for log2 fold changes and p-values. Significant genes were filtered by adjusted p-values using PAD-J less than 0.05 and a threshold for log2 fold change. Our second visualization would be the bar plot. We prepared this data for plotting by converting the DSeq2 results to a significant genes data frame and adding a column for gene IDs. We then selected the top 10 genes by base mean, and we also added a category column for up and down regulation using if-else statements and a condition of log2 fold change greater than 0. So what are upregulated and downregulated genes? These are genes that have increased expression, like a cell creating an abundance of RNA or proteins in response to a stimuli. And this would basically be vice versa for downregulated genes. So now that we had prepared our methodology for our volcano plot and our bar plot, we can now visualize the data and discuss the results, which will be handled out by Harrison.
Okay, moving into our results, our differential expression analysis revealed significant changes in the Arabidopsis transcriptome upon lead exposure. We identified dozens of genes showing statistically significant expression differences compared to the control group. Broadly, the affected genes fall into key functional categories: stress responses, metal transport and homeostasis, hormone signaling, and cellular detoxification, which aligns with the expectations for heavy metal stress. So this volcano plot visualizes these results. We're plotting the magnitude of change, log 2 fold change, on the x-axis, where the log 2 fold change represents the ratio of gene expression between our two conditions on a base 2 logarithmic scale. This means a value of 1 indicates a twofold increase in expression, a value of -1 indicates a twofold decrease, or half the expression, and a value of 0 indicates no change. This is plotted against the statistical significance, minus log 10 adjusted p-value, on the y-axis. As you can see, there's a clear distribution of genes that are significantly upregulated and downregulated, beyond our significance thresholds. This allowed us to pinpoint key candidate genes with substantial changes, which we have listed the top 10 differentially expressed genes here on the right. Next, this bar plot focuses specifically on the expression levels of these topmost upregulated and downregulated genes. You can observe consistent and pronounced shifts compared to the control samples. For instance, among the upregulated genes, we see ATAF1, a known stress-responsive transcription factor, and FRO2 involved in our iron uptake. Conversely, downregulated genes included developmental ones. We also see the LIA protein as very upregulated, which is no surprise as it is responsible for protection against various stressors. Finally, the table here on the right provides a summary of these top differentially expressed genes, including their functions. This reinforces the diversity of pathways impacted, ranging from actin remodeling, ADF11, and metal transport, FRO2, to flowering regulation, AGL28. The significant impact on transcription factors and metal homeostasis genes supports the idea of widespread regulatory changes triggered by lead. So the overall picture from the results is a clear systemic response to the transcriptomic level, an upregulation of defense and stress pathways coupled with a suppression of genes related to growth and development. Moving on to the discussion, we ask, what do these results imply? So our analysis indicates that Arabidopsis mounts a coordinated genetic response to lead exposure. Consistent with previous research on heavy metal stress, we observed the upregulation of known stress-related genes like the transcription factor ATAF1 and the iron transporter FRO2. This suggests the activation of defense mechanisms likely related to oxidative stress and maintaining metal balance. In contrast, the downregulation of key developmental genes involved in flowering development and ETC1 and trichome patterning points towards a resource allocation trade-off. Under toxic conditions, the plant appears to prioritize survival and stress mitigation over growth and reproduction, which is a well-documented phenomenon. An interesting finding was the upregulation of flowering time regulators like constans and AGL28. While perhaps less expected, this could hint at lead stress disrupting or reprogramming developmental timing signals, something observed under other environmental stressors. Lastly, it is important to briefly mention a limitation in our project. We utilized simulated replicates for this initial analysis due to experimental constraints. Replicates are essentially measurements taken from multiple different samples of the same species under the same experimental conditions. These measurements are crucial to account for any natural variations between individuals within their treatment groups. Therefore, while the trends are strong and informative, these results should be considered somewhat exploratory, providing excellent candidates for validation rather than definitive proofs on their own. And finally, due to the large size of our raw sequencing data, we optimized our initial processing. We compared a Python-friendly workflow utilizing the efficient star aligner with standard R bioconductor methods for alignment. The star-based pipeline proved more computationally efficient for handling this scale of data. Importantly, this allowed us to leverage star's speed for alignment before easily importing the resulting gene counts into R for the differential expression analysis using bioconductor tools. And next, onto our conclusion.
All right, to conclude, we should go over the summary of our findings. The summary of the findings are a few bullet points as follows. Pollution alters plant genes related to stress and mental homeostasis. So we can see that genes like ATAF1 and FRO2 were upregulated, and these are due to detoxification and iron regulation, which is related to the stress and metal homeostasis. Um, Developmental genes were suppressed due to the presence of lead. Lead exposure prompts a reallocation of resources to survival mechanisms. So it is more challenging for plants to be able to survive in these conditions, in part due to growth genes that are suppressed and more survival, such as stress related, are increased. This matters because by looking into this research, it supports conservation and land use policies. I think the findings showing that there are upregulated, downregulated, and even different genes that are upregulated that shouldn't be upregulated. This is an important part of needing to support conservation, since increased pollution causes plants to undergo more stress. It also provides biomarkers for pollution stress in plants. Now we know some biomarkers, and it also helps predict long-term ecological impacts, which we can see through upregulation of certain genes and suppressing of others. So we have our references, and there they are. Thank you for listening.